I'm Armelle Pindon. I'm the Chief Scientific Officer of Myhre Syndrome Foundation. So welcome to this again webinar. It's a Q&A. So the purpose is really for you to understand what we do, and at the same time have the opportunity to ask as many questions as you want, and I will try to answer it. Anything I say during this webinar, it's not as clinical advice. It's really to explain you the basic biology behind the strategy we have. So I'm going to share, hopefully, just the presentation. Is that okay?
Yes, that's perfect.
Okay. So for today, I would like to explain a little bit more. I don't know if you saw that we announced let's say the winner of the RFP. So the request for proposal that went out in the organization in late 2024. And after the whole evaluation by us and the scientific advisory board, the project was selected. It was the project on FAP CAR T cells. So it's an immunotherapy for Myhre syndrome. The main leader is Dr. Isabelle Richard from there's a typo here, from the Genethon Paris. And the co-PI is Dr. Valérie Cormier-Daire from Imagine Institute and the hospital. Kate, you didn't check you see the results if you don't check my thing. That's
No worries. We understand. You're multilingual. We get it.
So that's so from the hospital Necker Enfants Malades in Paris too. So those two PI are in fact really recognized in France. Isabelle Richard is the director of one of the main group or laboratory at the Genethon, and the Genethon has already I think 6 treatments on the market. Basically, they do gene therapy, but also other type of treatments. And Valérie Cormier-Daire, who's the one who discovered, in fact, the mutation SMAD4 and Myhre syndrome that made the link between that identify the mechanism of Myhre syndrome, that it's 2 mutation in the SMAD4 gene. So she's also quite famous, and she just had an award, a very prestigious award in France from the Institut Pasteur. So we can be proud that those 2 people are working with Myhre syndrome. So the CAR T cells. So I'm going to explain a little bit more what we do with this therapy. So we chose to invest in that technology because it's already in place for cancer therapy and is now adapting for cardiac fibrosis and muscle fibrosis and even other diseases linked to fibrosis. So starting from the beginning, scientists answer our request. So I just told you the RFP, and brought their project to adapt the existing technology, especially for fibrosis in Myhre syndrome. So it's really interesting how the discoveries in one area, which is cancer, is again one more time open possibilities for a completely different field. But it's not the first time. So the fact that it's already in place and there are already patients treated with that technology comforted us in the fact to choose this one because it's less risky for us. Although its CAR T cells are still really very advanced cutting edge technology. So for today, we are going to be looking at how the scientists are trying to use the CAR T cells and actually reverse fibrosis. The problem is there are no effective treatments out there right now for the fibrosis, except for cystic fibrosis, but that's a very specific type of fibrosis. So finding new ways to stop or even reverse this scarring is critical. So that's bring us to the world of CAR T cells therapy. So you've probably heard of CAR T cells. I mean, you see it even in the news to fight certain blood cancers, but researchers now are kind of figuring out how to adapt this technology to go after other type of cells than the tumor cells. For example, those activated fibroblasts or different sources of myofibroblasts that I explained you last time in explaining the fibrosis, those cells that goes into overdrive and trigger the fibrosis. So okay, so just to back up for a second, just to clarify. So CAR T cells basically involve taking a patient's own T cells, which is a type of white blood cells that's part of the immune system, and genetically modify them to target specific cells. So the T cells are going to recognize the cells they have to kill, and then destroy them, of course. But first they have to recognize the cells, the targeted cells. So you're basically giving the immune system a customized weapon to go after a very specific target. In this case, they are engineering those T cells to hunt down and destroy those activated fibroblasts that are causing all the trouble in the trachea, aorta, maybe gastrointestinal, we don't know, but a lot of fibrosis that is triggered in Myhre syndrome. So they are like those cells are really like the bad guys, and you need a target to get to them. So here it is. So that's naturally presents on those activated fibroblasts, something those engineered T cells would recognize. So the search was on for endogenous targets, and that's where they stumble upon something called FAP. So that's fibroblast activation protein. And now there are quite advanced research using this FAP as target to destroy activated fibroblasts, for example, in cardiac disease, in glioblastoma, or other tumors. So before we get into the details of FAP, I want to make sure you are all following along here. So just interrupt me and ask me questions if you don't understand what I'm talking about. So we've talked about the need for new therapy in Myhre, especially for fibrosis. And these potential CAR T cells to target those scar tissue producing the scar tissue produced by the activated fibroblast or myofibroblast. But finding that suitable target on those fibroblasts is really crucial. So it's like finding the, how do you say that in English? Achilles heel of fibrosis. Sorry for my French accent. We say Achilles in French, but
Achilles.
Achilles. Okay, thank you, Kate. So that's what we are looking for, in a way really to specifically target those activated fibroblasts. And what we want is that this target is not present on other healthy cells. You can, so the purpose is really to potentially develop a very precise and effective therapy. So here we are. So the first thing the researcher who applies, so Isabelle Richard and team, and Valérie Cormédier team, the first thing they did before applying to our RFP is that they checked that the FAP protein this fibroblast activation protein was present in the Myhre syndrome mouse model in different organs, and compare it with the amount of the FAP in mice that don't have Myhre syndrome. And in fact, they found that the FAP was significantly higher in the Myhre mice compared to the healthy ones. Okay, so that suggests that the FAP is indeed linked to Myhre. So it means that from the beginning, before starting any project, you need to be sure that what you are going to target is overexpressed in Myhre syndrome and is relevant for Myhre syndrome. So that was done, and then they applied to us. And what is interesting about the FAP, so the fibroblast activation protein, is that it's heterogeneously expressed on tumor cells, but consistently expressed by endothelial cells and pericytes and the different cells that are transformed during the process of fibrosis. So it's not only on fibroblast. It's, if you remember, last time when I explained the fibrosis, is that you can have epithelial cells that go through this mesenchymal transition, endothelial cells, and in Myhre, we have endothelial and epithelial that go through there. You have also smooth muscle cells, adipocyte, but that I don't think it's relevant for a lot for Myhre. But on those cells, when they are transformed, not when they are healthy, when they are transformed, you can see the expression of FAP. So that was really also very important. So the FAP has limited expression or presence in healthy tissue and vessels, but it's really present in the case of Myhre. So in the project, they are going to take the next logical step, and they are going to engineer the T cells to specifically target the FAP. So if we go back to this one, so they will have those CAR T with the FAP, which is in green, that so they are going to build this. And those CAR T cells that have the FAP are going to be able to recognize the fibrotic cells that have the FAP. So that's the recognition of FAP, and that's the FAP. And then they can start the process of destroying the cells. So it's not 100% safe. There is no treatment that is 100% safe. Even aspirin has some side effects, but they are trying to really decrease any potential side effect that can have this technology. And the safety is really a priority in any treatment you discover or you develop. So we know that those the FAP CAR T cells had a good track record in previous cancer treatment studies, showing no serious side effects without any signs of toxicity. So that's a pretty good safety profile. And what Isabelle Richard already did is that she has produced some FAP CAR T already and tried the safety in non-human primates, in monkeys, and the safety was quite good. So. But that's the study was on short term. So now, what about the long-term effects on having these engineered T cells in the body? And we can refer to the treatment that are now in people. There are now people going around with CAR T cells in their body from cancer and clinical trial in cardiac fibrosis. So what we are looking now is in the long term, what's happening to people, because they have those CAR T cells. They are designed to be this little warrior. What if they stick around and cause problems later on? So the persistence of these engineered T cells is really something the researchers are very aware of. And having these powerful cells circulating indefinitely could potentially lead to unforeseen issues down the road. So what's the solution, and how do you control these tiny assassins? Well, they are going to so there are different options that the team of Dr. Richards is working on. So the first one is to use mRNA to create temporary CAR T cells. So the level of control and precision becomes really higher. So what they could do, and what they are going to try to do during the this project is to so that's the chimeric antigen receptor. So that's going to recognize the FAP. What they are going to try is to induce that with putting the mRNA of that, so not the really expressing definitely. They are going to instead of taking the T cells out, modifying the T cells with putting the CAR inside the cells, grow the cells, and then re-inject them in the body. They try to put directly the CAR gene transfer with mRNA. So you heard about, there's now a lot of development of mRNA treatments for different type of diseases, and the mRNA is transient. So if you package the mRNA for the CAR gene transfer in, for example, liquid nanoparticles, and infuse it in the whole body, they are going to they need also to have another target for the T cells. They are going to be expressed in the T cells, but just for a short period of time. So you will have the CAR T cells in your body, but for a few 3 months instead of, or even less, instead of having them for a long period of time. So that would be one way to control. The other way is to have the other approach is to have a kind of switch off, in fact, on the CAR T cells, so that you can, whenever you want, destroy those T cells that have the CAR gene, the FAP CAR gene, for example. So there are different levels of controlling, like a switch on-off. Either you put it from the beginning and you know that it's going to be transient, so not for a long time, or you can have this switch on-off on the cells. And when you see that the patient starts to have side effects, then you kill those T cells, those particular T cells, the CAR T cells, so that everything stops. So that's two ways that they are going to work on. Basically, they are going to focus more on the transient expression, so that we can use it when the patient start to have really a fibrosis that is worrying, and we can use it just for a short period of time and then go back to normal. Or we can use it several times, because the way she wants to put the CAR gene transfer, the FAP recognition, is not via viruses, it's via the lipid nanoparticles. So you can do multiple injection of the RNA in the body. So that's just to explain the way we are addressing fibrosis with this project. It's a long project. It's a 3 years project, because it takes time to produce the CAR T, to have the proper, if I go back on that, so to have the right chimeric antigen receptor takes time too, the proper CAR T cells. She's using now the 4th generation of CAR T cells production. So that takes time, and then she's going to work in collaboration with the team of Valérie Cormier there, and they are going to really go deep in the characterization of the fibrosis linked to Myhre. Because up to now, we don't know a lot about the mechanism of fibrosis in Myhre. We know that there is tracheal fibrosis or other type of fibrosis that are life-threatening, but we don't really know exactly on which cell type we can observe it. So that's also the part of this project. And then they are going to infuse the CAR T cells and evaluate the dose and the regimen that they should use in human. So it's really it's a 3-year project, but it's going up to ready to go in clinical trial. So there will be the PK/PD. So that's that means the kind of toxicity study, safety study included, and establishing the dosage of either just the chimeric receptor. If we go for a direct infusion of the CAR gene transfer, or the proper amount of CAR T cells to reinject. So do you have any question? I'm going to stop sharing.
Yeah, I have a question, Armelle, which I already asked you before, but I think it'd be good to ask here because I imagine other people might have the same question. So like, fibrosis in the case of Myhre is just excess scarring, right? It's not like a new process. It's just too much of a process that the body already has. And so, and that scarring is important for, wound healing, and it happens all throughout the body and such. So if we target that, is there the chance that we're going to impede this natural process and create some side effect of the body not being able to heal?
Yeah, exactly. So that's one of the issue that's is taken into consideration in that project, is that we need to have a switch off in case you see that it goes too much and not goes and interfere with natural wound healing. So that's one thing. And the other thing is that we can in fact modify also the CAR T cells to be specifically directed to the type of fibrosis, and for example, we can direct to endothelial cells in addition to the FAP. So we can put 2 genes instead of 1 and target exactly the proper fibrosis or wound healing compared to another one. So that's also another option. But mainly the best way is to have this switch on off, or to have this transient, which is what they are going to work on. But you can also improve the specificity and go, for example, in not for the wound healing of the skin, but go more for the fibrosis around aorta. But that's not really in the project now. That can be a following step. But the thing is that the transient option is the easiest one, and the one that is used or developed now. Did I answer properly your question?
Yes. Although I like the second option better than the transient option. I worry about the practicality of the transient option. Like, would someone keep this off switch with them, like as an EpiPen? Like if they got a serious injury, would they hit themselves with it and then be able to heal from this injury, or like how does that work practically?
No, it's not. No, it's no, it's a treatment that you need to go to the clinician to get the treatment to switch off. But what she's working on is, I mean, it's a whole team. What they are working on is the transients. So just having the treatment, for example, if you have really tracheal fibrosis very severe or liver fibrosis very severe, you will receive the treatment. It would act for 2 months. They need, I said 2 months, that can be 6 weeks or 12 weeks. I don't know exactly because we didn't try yet. But that would be really for the time period. And for the time period, you stay in an environment where you don't go hunting at that time. But that's, I think it's the safest way to go. And that's the option they are choosing in the project, to go for transient. And it has several advantages to go in that direction, is that you also reduce the cost tremendously. And the time to prepare the CAR T cells is also totally reduced, because you don't need to take the T cells from the patient, work on them in the lab to modify them, and then grow them, re-inject them. So that takes time. So if you can directly infuse the CAR, so the FAP recognition, in the blood with lipid nanoparticles, then it goes, the whole process goes way faster, and it's way cheaper.
Thank you.
I have one question. And maybe I
Oh, sorry, you're cutting.
missed it, but are we talking about, can you hear me?
Yes, now, yeah.
Okay. Fibrosis, like if he were to, like if they were to get a really severe cut, like on the arm fibrosis, are we talking about like fibrosis like around the heart?
Yes, no, not for cut on the arm. Not skin fibrosis. I mean, it's not really called fibrosis in that case. It's an over scarring. No, it's really targeted for the heart, aorta, tracheal, liver, the fibrosis that are really life-threatening.
So you were saying the first part was looking at the fibrosis was, I'm sorry. So you said the next step would be to look at the fibrosis of the aorta, the second
No,
second part of this?
No, it's going to look for all the target is really the last part. So to trigger the fibrosis, you can start from the fibroblasts, the endothelial cells, which are around the vessels or the aorta, the epithelial cells. You have smooth muscle cells that are around the vessels also. So what we do in the foundation is that we focus a lot on vessels. So, for example, for our preclinical platform, one of the model is the microvascular endothelial cells from the brain. So we try to focus on those type of cells. So this CAR-T FAP is going to recognize the cells that overexpress FAP. What we do now with the next generation of CAR-T is to recognize one, so for example, FAP, that's the point, and try to have another one that can recognize more specific cells. So that will be all the FAP, but we can add something to recognize also specifically endothelial cells or epithelial cells, so that you are going to focus more on one type of fibrosis rather than the others.
Okay.
But that's for example, if you go for endothelial cells or epithelial cells, then it's vessels, aorta, heart, it's all of them. It's just not skin.
Okay. Okay. Awesome.
So that's a huge investment for the organization. But it's I think it's a safe treatment because it's on the market already for other application, and it's targeting really the main issue, the life-threatening. issues in Myhre. The thing with the with infusing directly the CAR gene transfer, so the FAP, for example, with lipid nanoparticles, is that now you can modify the lipid nanoparticle to enter the brain. So that might be also a development after, when we have the CAR T cells for fibrosis. We can also try to see if there are some cells that's in the brain that would express the FAP. And then if we use the proper vehicle with the liquid nanoparticles that envelop the mRNA and have this special label that will bring it through the brain, we can also foresee to use that in the outside the brain, not really inside, but it will never go inside the diencephalon, for example, but we can also use it around the brain, because they are using it now for glioblastoma. So that's also an option, but that's really for the future. It's not for now.
I have a question, Armelle. At what point would you see that someone, once it's developed and it works, at what point would you see that it would be used for a patient?
Well, when there is a level of stenosis that is dangerous, or for now we need to do clinical trial. We need to try in patients and see, maybe in very low doses, is it going to improve the general fibrosis that can be in the gastrointestinal, for example. We don't know, because there is so little study on the patients. We don't really know what's happening in gastrointestinal. We don't really know what's happening in the liver. So even in the endometrium. So that's well, we'll see. The first step is to build a tool and to use it in extreme in the cases that are really life-threatening. That's, I think the FDA would agree to do that. And then we can see, after, when it's agreed for that, then we can try to use it in lower regimen, lower dose, and see how it works on the everyday life, if it can release from other symptoms. But the first application would be really when you have something, a fibrosis that is dangerous.
And you did mention also that there would be sort of different ways of targeting different kinds of cells. And we might, start with one early and then do another one after that. Would the FDA consider these different targeting mechanisms as being different treatments that require their own clinical trials?
I don't know. If it's a different mRNA, then it's a different treatment.
That's expensive.
But in fact, then you design a protocol from the beginning that is a kind of it's an umbrella protocol. It's not the basket where you have different application with only one treatment, but there are some clinical trial protocol where you can have a patient going from one protocol to another with different treatment. So then the FDA you wouldn't have to go back to the FDA and do another application and everything, but you need to design that from the beginning when you do your the protocol for clinical trial, so that the patient can go from one box to another and try a different treatment or a different dose, or so that's the clinical trial design, especially for rare disease, are quite dynamic now with the FDA. And with the EMA also, even more in Europe, you can do way more things. So you can really you just need to think about that before you apply, and you talk with them, and they are going to now they really agree that you have dynamic protocols, where you can modify the protocol during the clinical trial.
Right. So would these be done all at the same time, or would a future mRNA guide be considered like an extension of the same trial if done later?
It depends on the first research. If we have the whole preclinical data ready at the same time to go for the different type, or if we do the preclinical developments one after another. First, only the FAP, and then something else added to the FAP. So if you do that in parallel or all together, then it's then you can present in the clinical trial in parallel or one after another. But for now, the project does not involve all the different cell type. That would be for a later. For now, what they present in the project is really the FAP in Myhre with transient application.